Hello everyone, I'm Dr. Divya Jain, Department of Botany, SD College, Ambala, Kent. In this tutorial, I'm going to introduce you to DNA damage and some repair mechanisms that the cells have evolved to protect themselves from mutations. The tutorial is divided into two parts. In the first part, we will study the causes and the types of DNA damage. And in the second part, we will focus on some mechanisms the cells have evolved to repair the damaged DNA. First of all, you should know what actually is DNA damage. Any change in the physical or chemical structure of DNA, or both, that is, both in the physical or the chemical structure, that interferes with the replication and the transcription of the molecule is called DNA damage. That means any modification, any change in the physical or chemical structure of DNA, or both in the physical and the chemical structure of DNA that interferes with replication and transcription of the DNA is called DNA damage. Now, this change or damage, it may be caused due to exposure of DNA to chemicals or radiations, or these changes they may be spontaneous. As seen in the diagram, it can be a break in the single strand of DNA or both the strands of DNA. It can be a physical distortion due to a dimer formation within the strand or mismatch of bases in the molecule of DNA, or it could be the presence of AP site, which is a position from where the nitrogenous base has been removed, or there may be a mismatch in the strand of DNA. As you know, the two strands of DNA are complementary to each other because the nitrogenous bases of DNA they show specific pairing, that is adenine pairs with thymine and guanine pairs with cytosine and vice versa. If pairing does not follow this rule, it's a mismatch and can cause mutations. In addition to it, the bases may also undergo modifications. For example, very often you would see that cytosine loses its amine group and it forms uracil. Now, uracil is not the normal base of DNA and it won't pair with guanine, it will base pair with adenine, and in the cycles of replications, if this damage is not repaired, then the GC base pair would be substituted by the AT base pairs. So DNA damage is any physical or chemical change in the structure of DNA that interferes with replication and transcription of the molecule. It can be a double stranded break, it can be a single stranded break, it can be a physical distortion due to the formation of the dimers within the molecule, it can be a mismatch of the bases, it can be an AP site from where one of the nitrogenous bases has been removed, or it may be due to a modified base. And all these damages, if they are left unrepaired, they can cause mutations and they can ultimately lead to cancer. Well, students, we often correlate DNA damage with mutations. However, we need to understand the difference between the two. DNA damage normally refers to the injuries of DNA molecule that causes deviation from its normal intact structure, like we just discussed in the previous slide. If these injuries they are left unrepaired, they result in a mutation or they block the DNA replication. So, any change or modification in the DNA molecule is known as the DNA damage, while mutation refers to the permanent alteration of the base pair or nucleotide sequence of the DNA of an organism. So, if there is permanent change in the base pair or nucleotide sequence of the DNA of an organism, we refer to those changes as the mutations. Now, for example, when cytosine deaminates to form uracil, that means it loses its amine group, and when this amine group is lost, cytosine, it changes to uracil. This change is known as the DNA damage or modification, and if this damage is not repaired, it would cause mutation, which you know as substitution. Let's see how. In this diagram, you see the normal DNA has a GC base pair. Due to some external or internal factors, cytosine loses its amine group and it deaminates to uracil. Now, this uracil is not the normal base of DNA. Moreover, during replication, this uracil it will base pair with adenine. And in the next cycle of replication, adenine will base pair with thymine. And you see, now this is the AT base pair and this was the GC base pair. So that means this GC base pair, due to deamination of uracil, which is not repaired, and ultimately leads to the substitution of the base pair of GC to AT base pair. So, deamination of cytosine to uracil is the damage, while in subsequent replications, when this damage is not repaired, the substitution of GC base pair to AT base pair is the mutation. Now, the cells, they have mechanisms to fix DNA modifications or damages, but the mutations are the permanent status of the cell. Now, the question is why and how the DNA gets modified or damaged. You see, both exogenous and endogenous factors are responsible for the DNA damage. Exogenous factors are the external factors, and the endogenous factors are the internal factors. The major source of endogenous DNA damage or modification is the intracellular metabolism, which produces free radicals like superoxide and hydrogen peroxide. Living cells also contain reactive molecules like S-adenosylmethionine and betaine. These chemicals they add alkyl groups to DNA, and alkylation of DNA interferes with replication. The DNA bases spontaneously undergo hydrolysis, and this results in deamination or removal of amine group from the bases. The classic example of this is deamination of cytosine to uracil, which is not a DNA base, and how deamination of cytosine to uracil brings about mutation we have just discussed in the previous slide. As you know, DNA replication is a multi-step and a complicated process. Any error in replication and Any error in proofreading of the newly formed DNA strand can cause modification in the DNA molecule. Mostly, this error in replication and proofreading results in mismatch pairing of bases. The exogenous or external factors responsible for DNA damage or modification are UV lights, both UVA and UVB light, ionizing radiations, industrial chemicals, and elevated temperatures are the other exogenous factors which cause modification or damage to DNA molecule. So, all these factors in one way or the other modify the DNA and disturbs the replication process. Let's see what kind of DNA damage or modification is caused by these internal or external factors. Let's discuss one of the factors that is responsible for DNA damage, that is oxidation of bases. 8-oxoguanine is one of the most common DNA modification. This damage is caused by reactive oxygen species, also known as free radicals. These are produced as a result of intracellular metabolic processes. The reactive oxygen species oxidizes guanine to 8-oxoguanine by addition of an oxo group. You see, this is the oxo group to carbon 8 position of guanine. So, to the carbon 8 position, reactive oxygen species adds an oxo group and guanine becomes 8-oxoguanine. This 8-oxoguanine, it base pairs with adenine, and you know that the normal guanine base pairs with cytosine, but after the oxidation the guanine does not base pair with cytosine, it base pairs with adenine. And you see, now in the DNA strand, this guanine, it is slowly being replaced in subsequent replications with adenine. So guanine has been oxidized, and this oxidized guanine does not base pair with cytosine, it base pairs with adenine, and therefore cytosine has been replaced by adenine. This causes a type of mutation which you call as transversion, in which a pyrimidine, that is here cytosine, has been replaced by a purine, that is adenine. Another type of damage is caused by alkylating agents. These agents add methyl or ethyl groups to the bases, which result in mispairing of base pairs. For example, exposure of DNA to alkyl agents like ethyl methane sulfonate results in the formation of O6 ethyl guanine. Now, O6 ethyl guanine, it mispairs with thymine. This is the normal guanine, and after the alkylation, so here the ethyl group is added to guanine and it becomes O6 ethyl guanine. This O6 ethyl guanine, it base pairs with thymine and it causes mutations. because in the next cycle of replication, this thymine, it would base pair with adenine. So therefore, there is a substitution of GC base pair to AT base pair. Let us discuss another type of DNA damage caused by deamination of bases. Deamination of bases we have already discussed in the previous slides one of the examples. During the deamination process, an amino group is removed from the bases. We have already discussed how the cytosine it spontaneously deaminates to uracil. It loses its amine group and becomes uracil. Uracil is not the normal base of DNA and also it base pairs with adenine. So the deaminated cytosine becomes uracil, uracil base pairs with adenine and adenine base pairs with thymine. So in the subsequent replication, you will find that cytosine and guanine base pair has been replaced by adenine and thymine base pair. Similarly, if the amine group is removed from the nitrogenous base adenine, it becomes hypoxanthine. And this hypoxanthine base pairs with cytosine instead of thymine. So earlier, which was an AT base pair, it gets replaced by the GC base pair. Hydrolysis of bases is another type of DNA damage. DNA consists of long strands of sugar molecules called deoxyribose that are linked together by phosphate groups. So you know that the DNA backbone is made up of deoxyribose sugar, and these sugar molecules are linked together by phosphodi- phosphate group by phosphodiester bonds. Each sugar molecule carries one of the four natural DNA bases adenine, guanine, cytosine, or thymine. And together, the sugar molecule, the phosphate group, and the nitrogenous base constitute the nucleotides, and the adjacent nucleotides they are linked together by phosphodiester bonds. Now, the covalent structure of DNA is unstable in aqueous solution, though the chemical bond between a base and its respective deoxyribose, that means the chemical bond that is between the sugar molecule and the nitrogenous base is the glycosidic bond, and it is usually very stable, but sometimes it undergoes cleavage by water molecule, and this process is known as hydrolysis. Hydrolysis of DNA bases results in deamination of the bases, and it also results in depurination of the bases. So deamination means removal of amine group from the base, and depurination is the loss of purine from the nucleotide of DNA. So I hope it is clear how the hydrolysis of bases it brings about deamination and depurination. So these are the nucleotides of the DNA molecule. Each nucleotide consists of a sugar molecule, a phosphate group, and a nitrogenous base. And the adjacent nucleotides, they are linked together by phosphodiester bonds. Now, DNA, like I just told you, it is unstable in aqueous solution, and sometimes the water molecule, it causes the cleavage of the bonds that exist in these nucleotides. So, this cleavage can result in deamination of the bases. Like here, the cytosine, due to hydrolysis, deaminates to uracil, and uracil will base pair with adenine in subsequent replications, and therefore a GC base pair would be mutated to AT base pair. In this case, in depurination, after hydrolysis, guanine or adenine, that means the purines, they get removed due to the breaking of this glycosidic bond between sugar and the base. Now, as this guanine or adenine, it gets removed from the DNA strand, an empty base site is left in the DNA strand. This site, which does not have any nitrogenous base, is known as the AP site. Okay, so AP site, it can be apurinic site or it can be a pyrimidinic site. If purines are removed, it is apurinic site. If pyrimidines are removed, it is a pyrimidinic site. But usually it has been observed that the purines, they get removed at a faster rate than the pyrimidines. So now see what happens at this AP site. Let's say the two strands, they are replicating. The strand with the apurinic site, there, this is an abnormality, and a deletion would occur at this point because DNA polymerase would be unable to read this apurinic or AP site, apurinic or apyrimidinic site, and it won't read it, and a deletion would happen in the DNA strand, and deletion can cause mutations. That means if this site is not repaired, then the mutations will happen in the cell. Next major DNA damage is caused by bulky DNA adducts. These adducts are the major cause of carcinogenesis. Now, what is bulky DNA adduct? You see, DNA adduct is a piece of DNA, that means a part of DNA covalently linked to a chemical. For example, polycyclic aromatic hydrocarbons, like these, like aristolochic acid, 4-amino biphenol. So all these chemicals, they become covalently linked to the DNA molecule. And these molecules, they are present in tobacco smoke, or they also occur in cisplatin, a medicine used in chemotherapy of cancers. And these molecules, they become covalently bonded to the DNA molecule and form the bulky adducts. Like I just told you, these bulky adducts are the major cause of carcinogenesis because when a chemical binds to the DNA becomes damaged and proper and complete replication cannot occur, and the cell cycle becomes unregulated. It might lead to the death of the cell also. Let me tell you one very interesting observation here. It has been observed that the excess consumption of red meat, that is the meat that we take from beef, lamb, or pork, it increases the risk of colon cancer. And after the research, it has been reported that this increase in the risk of colon cancer is due to the formation of DNA adducts such as methyl cytosine and carboxyl adenine from the digestion of red meat. So I hope now you understand why your nutritionist they advise that red meat should not be taken in excess. Another important exogenous factor responsible for DNA damage is exposure to radiations. These radiations they can be UVA light, UVB light or the ionizing radiations like X-rays or gamma rays. UVA light causes free radicals, and as I told you earlier, free radicals cause oxidation of bases, and as you know, the oxidized bases don't show normal pairing during replication. Let's see how UVB light damages the DNA. See, when a DNA molecule is exposed to UV light, two adjacent thymine bases they become covalently linked together. That means if in a DNA strand there are two thymines that are existing adjacent to each other and they become exposed to UV light, these two thymine bases they will become covalently linked together forming a thymine dimer. Now this covalent linkage or the thymine dimer causes structural distortion of DNA. It produces kinks in the DNA molecule, and this structural distortion disrupts the replication. Let us see how. Now, if the replication is taking place and there is a thymine dimer in the template strand. Now, as the polymerase is moving along the template strand, bringing about replication, and as it encounters the thymine dimer, it slips off from the template strand, leaving a gap in the daughter strand opposite the thymine dimer because the DNA polymerase cannot read the dimer and it slips off the template strand. Okay, so it again rejoins the replication after crossing the dimer. So as it is moving on the template and it encounters this dimer, it slips off, and as it slips off, a gap is left in the daughter strand, but polymerase again rejoins. So it leaves behind this portion of the DNA that is not copied on the daughter strand and a gap is left in the replicating strand. And a gap, as you know, can lead to deletions or substitutions. Now, let's see what happens when the DNA is exposed to ionizing radiations. Ionizing radiations are caused by radioactive decay of radioactive elements, or they occur in cosmic rays. For example, X rays or gamma rays. These radiations they cause single strand breaks in the DNA molecule, or they may bring about double stranded breaks in the DNA molecule along with other damages and abnormalities. You will be surprised to know that every day around more than 1 million individual molecular lesions happen per cell, either due to endogenous factors or due to exogenous factors. Fortunately, by nature, the cells have evolved the mechanisms to detect the DNA damage and then to repair the damage. Several DNA repair mechanisms have been identified in both eukaryotic and prokaryotic cells. So, these are the damages that we have already uh, discussed, and these are caused by these factors: UV light, alkylating agents, reactive oxygen species, and these they are the damages that are the mismatches or the deamination or the modification of the bases or the breaks in the DNA molecule. But the cells are clever and they have developed certain repair mechanisms, and these repair mechanisms, mismatch repair or base excision repair, nucleotide excision repair, all these repair mechanisms we will be discussing in the next tutorial. Meanwhile, you can attempt these questions to find out how far you have understood the concept of DNA damage. Define DNA damage. How is DNA damage different from mutations? Name the two sources of DNA damage. Explain how free radicals are responsible for DNA damage. How do alkylating agents damage DNA molecules? What are bulky adducts? How are they responsible for DNA damage? What happens if the DNA bases are hydrolyzed? What is deamination? Explain how it is responsible for mutation. How do ionizing radiations damage DNA? And how is UVB light responsible for DNA damage? With this, we come to the end of this class. In the next class, we will learn some DNA repair mechanisms. Meanwhile, go through the concepts taught today carefully. Happy learning!